Welcome to Odisha NET Academy. Now I am going to discuss about YTH, Yeast to hybrid system. Yeast to hybrid system, this is one of the advanced technique to detect whether two proteins are interacting or not. Yeast to hybrid system. Okay, so before that you should have some idea about transcription factor. I think you all know what is transcription factor. Now this is the proteinous mostly and which regulate the gene expression after binding with your promoter region. So it mostly attaches to upstream region of the gene, that is promoter, and enhances or causes the gene expression. And it must have at least two domain. Remember, okay, one is DVD, DNA binding domain, which is attached to your promoter region, and one is PID, protein interacting domain, or you can say TA, trans activating domain, which interact with DNA, RNA polymerase. DVD interact with what? DNA. So each transcription factor must have two domains, and these two domains are derived from two different genes. That's why it is called trans-acting factor. Transcription factor is called as trans-acting factor. So now come to YTH. So in our last lecture, okay, so we have discussed about your FRET. So most of the steps are similar with FRET. So now come to YTH. So as similarly, whether we are going to detect whether A protein and B proteins are interacting or not. So like that, like FRET, you have to construct gene A, you have to construct gene B, then you have to take one suitable host, that is yeast, and modify the yeast. What type of modification? Now incorporate or insert GFP gene along with its promoter in the genome of yeast. Okay, and for expression of GFP there must be transcription factor, and that transcription factor is not present in yeast because GFP gene is its not its own gene. That's why. So you have to incorporate what? Transcription factor. How? Now the DVD gene of transcription factor of GFP will be fused with A gene, and PID or TA gene of transcription factor of GFP will be fused with B gene. Now these two gene fusion gene will be inserted into expression factor and produces hybrid 1, hybrid 2. And co-transfer, simultaneous transfer these two hybrid into modified yeast. So that's why it's called yeast to hybrid system. Now assuming that expression rates are equal, so your A and DVD fusion gene produces A fused with DVD. Similarly, B and PID gene fusion gene produces B fused with PID protein. So irrespective of AB interaction, who will come and attach to promoter? DVD. See, whether it is interacting or not interacting, DVD attaches to promoter. As what? DVD attaches with A, so A also come closer to promoter. Suppose case 1, interacting case. A and B are interacting. Now A come closer to B. So if A come closer to B, along with B, who come closer to promoter? PID, and recruit RNA polymerase and causes transcription followed by translation to produce GFP. Okay, all these things occur inside the cell, but I am not present inside the cell. I am unable to see what are the things happening inside the cell because I am outside the cell. So what you have to do? Now expose or keep the cell or expose the cell under fluorescence microscope and allow lambda or wavelength which can excite only GFP. So if you are getting green emission, that means green emission, that means GFP is produced. GFP produced means gene expression occur. Gene expression occur means what happened? A and B are interacting. Only when A and B are interacting, then only DVD and PI are come closer to promoter, and it causes what? Gene expression. Similarly, suppose A and B are not interacting. Still, DVD come and attach to promoter along with what? A, but A and B are not interacting. So PID is somewhere else. So due to lack of PID at promoter side, no expression. So when the cell is excited with lambda, so there is no emission. If there is no emission or there is no emission, so what is your conclusion? A and B are not interacting. So this is one of the in vivo technique through what you can detect whether two proteins are interacting or not interacting. Got it? And see here, so your DNA binding domain, this is AD, protein interacting domain. Only when both are interacting, then only you are getting gene expression takes place. So see here, suppose my protein of interest is bat and prey. So only when bat prey are interacting, gene expression takes place. So reporter gene getting what expressed, and you are getting what, some color, some light, like according to the nature of reporter gene protein. If bat and prey are not interacting, no expression, no questions of any light or any color, anything. Okay. So what are the drawback? Now same drawback as that of your prey. Already I have told you conformation drawback. That is what your differential expressional drawback. Okay. Another one is always fusion gene are not going to produce fusion protein. And here also pairwise expression can be studied only. If number of proteins are there, I cannot say which protein are interacting with what, which protein. Now come to this question, which of the following is incorrect about Yeast 2 hybrid system or screen? The incorrect thing, it is asking about incorrect. The Yeast 2 hybrid system uses the transcription of a reporter gene driven by Gulf transcription factor to monitor whether or not two proteins are interacting. Yes, this is used for the protein-protein interaction. That is correct. The DNA binding domain chimeric protein will not bind to offspring. No, always it binds to offspring whether the proteins are interacting or not. Because DBT always attaches to promoter present in upstream region. So the statement is wrong. If activation domain means protein interacting domain, PID. Activation domain chimeric protein interact with DNA binding domain chimeric protein, then only what happen, reporter gene will be transcribed. Correct. Disadvantage of method are that only pairwise interactions are tested. Yes, not interactions that can only take place when multiple protein come together, as well as a high false positive rate. So this statement is incorrect. So your answer is B. Another question, which of the following describes an advantage of Yeast 2 hybrid method for analysis of protein interaction? Okay, now go through one by one options. The assay works well for membrane bound protein, is it? The assay can screen for interaction partners of protein without the need of protein purification. Yes. Okay, here there is no questions of protein purification because it is in vivo. The assay only detects direct association between two protein, no indirect association. Okay, this is also not correct. The assay secretes protein, no questions of secretion here. So the best answer is B here also. Now come to another question. All these are previous year asked question. So in the Yeast 2 hybrid system, which of the following statement is accurate? A reporter gene is fused to activation domain. Reporter gene is not going to fuse with activation domain. Reporter gene is somewhere else. Reporter gene is fused to DNA binding domain. No. Reporter gene is not going to fuse with any other things. Reporter gene requires the presence of histidine in the growth medium. No. Have I told you what the growth medium requires histidine? Nothing. No. The reporter gene is expressed only if the tested protein interaction occur. Yes. Only when bat and prey or A and B or your tested protein are going to interact, then only expression of reporter gene takes place, because the interaction of A and B causes the interaction between DBD and PID, and PID comes closer to promoter region, causes the expression of your reporter gene. So this is the best answer. Clear everybody? So this is all about your YTH yeast 2 hybrid system. Okay. Thank you everybody.